Hello everyone. So welcome to this session on molecular drivers of the plant-bacterial interactions. I am Boris SZUREK from IRD. I work in Montpellier at the Plant Health Institute of Montpellier. I'm co-chairing this session with my friend and colleague, Jonathan.
I'm Jonathan Jacobs. I'm at Ohio State from the US.
So we have 6 speakers in this session, and one did not load his presentation. So I'm asking Huanbin ZHOU if you are around, if you are in the audience, if someone knows him, can you quickly upload your presentation at the preview desk? You still have time. Okay. Practical stuff.
So we'll be like other sessions. You can get up to the mic and ask questions, but please use the app by clicking interact. And the room that we're in is the amphitheater, so please use that as a space to ask questions, and we'll kind of track those questions in the chat.
Okay, so we will start now with the first speaker, but before this, we would like to say a few words about a friend and colleague who passed away less than 2 months ago, and we felt it was the good time to do this today, because if he would have been able to attend this Congress, he would have been here for sure. So I would like to ask Saul to come up and just share with us.
Well, thank you, Boris and Jonathan, for asking me to say a few words in the memory of Guido, our friend, that passed away prematurely on July 4, when he was 59 years old. So, yeah, Guido was not just a colleague; he was a very good friend of many of us in this audience. So it's an honor for me to give to say a few words about him, and I will probably get emotional about it. I'm still not used, as many of us, to the idea that talking about Guido in the past tense. So I had to write this stuff. So Guido was born in 1964 in Rome, Italy. By the way, he was a big supporter of Lazio. He liked football a lot. So he immigrated to Israel in 1983. He studied for his bachelor's degree at the Faculty of Agriculture, Food and Environment of the Hebrew University. He then completed a master's degree and a doctorate at the Weizmann Institute of Science under the guidance of Professor Robert Flohr. Then, in 1996, he got funding from the U.S. Israel Bard Foundation and moved to the U.S. for a postdoctorate under the guidance of Professor Greg Martin, first at Purdue University and then at Cornell University. So during his postdoc, he specialized in the study of molecular and biochemical aspects of interactions between plants and plant pathogenic bacteria. In 2000, Guido returned to Israel and established his research group at the Faculty of Life Science at Tel Aviv University in the Department of Plant Sciences, which later became the School of Plant Sciences and Food Security. He progressed through the academic ranks to full professor and was a senior and significant member of the faculty's academic staff and research team. Between 2018 and 2022, Guido served as the head of the School of Plant Science and Food Security. And recently in this year, he was appointed head of the Cereal Research Institute at Tel Aviv University. So Guido's research focused on interactions between plants and plant pathogenic bacteria, when he took a dual approach. On the one hand, he studied the processes leading to the activation of immune response in the plants. And on the other hand, he aimed at understanding the mechanisms that allow pathogenic bacteria to colonize the plant and cause disease. In recent years, Guido started a new line of research studying the interactions between the plant microbiome, the environment, and how they affect plant disease. Guido collaborated with many researchers abroad and in Israel, including myself. I want to mention a particularly prolonged and highly fruitful collaboration was with Professor Greg Martin, who, as I mentioned before, guided him in his postdoctorate, but they continue working and they joint work, they continued for many years, led to the publication of groundbreaking studies. Guido was also a great teacher of courses in the area of plant microbe interactions. In addition, he had many friends in other research institutes in Israel, including in the Faculty of Agriculture, to which I am associated of the Hebrew University, in which, as I mentioned, he did his undergraduate studies. And through the years, Guido greatly contributed to the academic activities of our faculty, including as member of PhD committees and examiner of master's students. In recent years, Guido worked to promote research collaborations in the plant pathology field between researchers from Italy and Israel through the mediation of the Italian embassy in Israel. At the peak of the project, he organized a joint one-day workshop in Israel with the participation of researchers from both countries. Unfortunately, he did not get to participate in it. About a week before the workshop, Guido entered a state of coma and artificial respiration. At this point, it was already clear that there was no way back, and the question arose whether to hold the workshop or not. At the end, we decided to hold it, also after consulting with his family, mainly because we knew how important it was for him and how much he worried about it happening, even at the time that he was already hospitalized. The workshop was held on July 3, one day before Guido passed away. It seems that Guido was holding on to make sure we were up on this task. Guido was an exceptional scientist and an exemplary mentor to young researchers and to his students. Many of them went to academic careers in institutions in Israel and around the world or integrated into the biotech industry. At the personal level, Guido was just an excellent person. All that knew him know that he had a good and clean character. He was extremely modest and always willing to help students, new scientists, and colleagues. Always with goodwill, patience, and above all, a huge heart. I can testify to this firsthand, as he helped me a lot at the beginning of my career, and also when I was an established researcher. The word competition was not present in Guido's dictionary. Another feature of Guido is that he was a great partner for international conferences, as many people in this audience can testify. This is just one example. So, with all the sadness of his death, I'm glad that I had the privilege to be his friend, to spend many moments with him, to collaborate with him, and especially to learn from him. I want to mention also that Guido was also a devoted and loving family man, and particularly of his lovely daughter, Shira. In these days, I'm working with Guy Sobol, who is Guido's last PhD student, in the preparation of a paper that resulted from collaboration of our groups about one acidophorus type 3 effector. As said, it is hard to understand and accept that Guido is no longer with us, although he is and will always be in the heart of many of us. Thank you.
Thank you, Saul. So we were speaking about Guido SESSA, if people that don't know him. Now we can welcome our first speaker, Alice Boulanger from Toulouse, INRAE, Université Paul Sabatier.
So, good morning, everyone. It's a real honor and pleasure to be here to present the work that we performed the last few years. I have to say that I'm a bit intimidated by the size of the room. It's the first time that I give a talk in such a big room. So, my name is Alice Boulanger. I'm working at LIPME in the group of Laurent Noël. And our group is interested in the infectious strategies of Xanthomonas. So I will quickly present our pathosystem. So we are working on Xanthomonas campestris pathovar campestris, or XCC, that cause black rod disease on Brassicaceae. And you can recognize the disease by the typical V-shaped symptoms that you can observe on leaf of crops or the model plant Arabidopsis thaliana. This bacterium has a complex disease cycle. It can survive on leaf surface, and when conditions are favorable, it will enter plant tissue, either by wound or natural entry points that are hydatodes. And from this point, it will colonize vasculature and multiply in xylem vessels, but it also can escape from the xylem vessels to colonize mesophile. And when the bacterium becomes systemic, it can be transmitted to seed. And as we have seen this morning, this is a big issue for seed industry. So the virulence factors of this bacterium are really well described. However, we don't know all genes that are really important for the colonization of each compartment that the bacterium will encounter through its cycle. So to have a better idea of this repertoire of genes, we have decided to perform high-throughput fitness analysis using RBT&SEC, and this was done in collaboration of Adam Dodgebauer's group and Jennifer Lewis' group in Berkeley. So briefly, what is RBT&SEC? RBT&SEC relies on a complex mutant library where each strain is barcoded. So if you sequence and count the copy of the barcode that you have in the population before the treatment and after the treatment, you have an idea of the contribution of each gene in the fitness of the bacterium in the conditions that you have essayed. So we did that in the first endophytic tissues that the bacterium encounters, the hydatode. So our hydatodes are located at the leaf margin, and they are involved in a guttation process. And it's a really good place for a vascular pathogen to start an infection, because as you can see on this picture, there is many pores located at hydatodes, and under them you can find many xylem vessels branches. And as you can see here, XCC can localize to the hydatode pores and as a successful colonization of epithemal tissue under these pores. And you can see here, at really, really early step of infection, bacteria between epithemal cells. So one first thing that we had to check before doing this RBT&SEC is the bottleneck. You don't want, it's really challenging to do RBT&SEC if only one or two strains enter your tissue. So to do that, we count the different barcodes that you can have in each hydatode sampled. And we found an average of 35,000 barcodes, different barcodes in each hydatode. So that means that we can do our RBT&SEC in hydatode, and so we did. And here are the results that we obtained. So 6 days post-inoculation, you can see that there is plenty of genes that are in red here, along XCC genome that seems to be affected in hydatode colonization, and we identified 181 genes contributing to XCC fitness in this condition. So if we take a closer look to the functions encoded by these genes, as previously observed for other pathosystems like Agrobacterium, Dikea, or Pseudomonas, there is a big part of these genes that are involved in amino acid metabolism, purine pyrimidine metabolism, or carbohydrate. But we also have genes that are involved in signaling and regulation, and I have the feeling that these ones are of the main importance. Virulence, stress response, transport, but also a big part encode for protein with not non-function. meaning that RBT-NSEC is a good tool to identify potential new virulence factor. So we have decided to choose 12 candidates representing the diversity of function that we can find here. And we did clean deletion of this mutant. And the first thing, as pathologists, we did was to look at the role of these genes in virulence of XCC. So we directly inoculated our deletion mutant in the central vein of cauliflower leaf and scored the symptoms appearance over time to get the disease index. And one thing that was obvious for us was that there is not always correlation between involvement in fitness and involvement in pathogenicity. As you can see here, there is 4 genes that show a decrease of fitness in idatod, but do not show any decrease in pathogenicity compared to the wild type that you have here. However, there is other genes that show a decrease in pathogenicity. These 2 genes are impaired in the type 3 secretion system, so it's a control, because the type 3 secretion system is a major virulence factor of XCC. And among these genes, there is some that in fact show a gain of fitness in idatod. So we identified 13 genes with a gain of fitness in our idatodes. And if we look at these genes, 10 of them encode for really well-known regulators of the bacterium. And you have, for example, the 2 major regulators of the type 3 secretion system, ARPX and ARPG. So why do we see this gain of function for these factors of virulence? It's because in our condition, you can have cross-complementation between the mutants inside the library. So if we take the example of RPX, an RPX mutant will no longer produce a type 3 secretion system, and in our library will behave like a cheater and benefit of the production of the type 3 secretion system of the other strains that are wild type for this virulence factor. So in fact, this gain of fitness represents the fitness cost of the production of the type 3 secretion system. We also identified 3 genes encoding for proteins with not non-function, and one of them show a decrease in pathogenicity. So we have decided to characterize deeper the role of these genes that we named PAC-R. So these genes is conserved among all plant-associated proteobacteria. If we look for domains, you will not find anything except a DUF domain. And if you look for the predicted structure using AlphaFold, it will look like other genes with the same DUF domain, but nothing that was previously characterized. So we phenotype our mutant, and the first phenotype that we observed was that it looks like this mutant do not produce as much EPS as the wild type. So we quantified that and confirmed that our mutant do not produce as much EPS as the wild type or the complemented strain. We also observed that this mutant do not produce amylase activity as much as the wild type. And we also observed an hypermotile phenotype for our mutant. So here you can see the motility of swimming motility of our mutant compared to the wild type. And when you overexpress PAC-R, because when we complement our strain, we put the gene under a constitutive promoter, so it mimics a bit an overproduction, you decrease the motility, strongly the motility of the mutant. So seeing a decrease of EPS, increase of motility, of course, we have decided to look at biofilm formation. And we were able to observe that the biofilm formed by PAC-R was really disturbed in comparison to the wild type, especially at 72 hours, or the complemented strain. So we have this pleiotropic effect, but our big question was why. So to try to have a lead on that, we have decided to perform RNA-seq and compare the expression of XCC genes in our 3 strains and identify 153 genes that were differentially expressed. And some of them recapitulate the phenotype that we observed. Like, for example, you have a decrease of expression of 7 genes involved in EPS, and you have induction of 30 genes in chemotaxi and 33 genes in flagellar motility. And we were really surprised to find 15 genes that were upregulated, coding for cyclic DGMP regulators. And so that's why we named these genes PAC-R for pathogenicity and adaptation through cyclic DGMP regulation. So it's already really well known that cyclic DGMP is an important factor for the regulation of many cellular processes in many bacteria, and also XCC. But if you look at the two component system or the regulator that are directly or indirectly associated to cyclic DGMP, all the one circled in red show an increase of fitness in our RBT and SEC analysis. And so now the question is where we can place PAC-R in this scheme. So we would like to check first, of course, if the cyclic DGMP level change in a mutant background. We would like to know how PAC-R is regulated, how PAC-R regulates genes, and does it bind DNA, RNA, interact with other proteins, and so on. So this work was published, but I have just a bit of maybe of time to talk about what we are also doing on other plant compartments. So we continued our RBT and SEC analysis, and we performed the same fitness screens on xylem vessels and mesophyll. And I don't have time to go deeper in the results, but what I can say is that we have distinct genes that seems to drive the colonization of these different compartments. For example, we have 38 genes specific that seems to be specific to idatod, 34 for xylem vessels, a bit less for mesophyll. And also one interesting thing is nearly half of the genes that we identified in our in-planta screen do not have any phenotype in our in vitro screen when we do RBT and SEC in artificial media, meaning that this gene seems to be really specific to the in-planta condition. So with that, I would like to thank all our group members, but especially the brilliant PhD student who did the work. So Aude Serruti is a former PhD student who did a lot of work on characterization of idatodes and colonization by XCC. Julien Luneau performed all the work on fitness analysis in idatodes. And Thomas Quiros-Monens performed the work on xylem vessels and mesophyll fitness analysis, and he presented a poster. So we have the number, so I encourage every one of you that are interested in this work to go to discuss with him. We also have Corinne Audran and Brice Charles that present posters about Tal's effector. So if you are interested in this topic, please go to see them also. And I'm waiting for your question.
So if you could please come to the microphone to ask a question. We have one already in the chat. So the first one is PAC-R structure or domain organization known? Which I think you handled that with the Duff, but I'd be curious to hear about the structure and domain of this. And then can this help formulate hypotheses about its role in direct interactors?
So yeah, we looked at the prediction of the structure, as I said, using AlphaFold, and look if we can find something looking like that in DALI server. We didn't find anything, but if you cut the protein in different domains, there is one domain that looks like TRA-E, a protein that is known to bind DNA and bind type 4 pilus, and that is really a main protein involved in the conjugation process. So transfer DNA through the type 4 pilus. So maybe PAC-R can bind DNA, but we have to test that.
Hello, Elise. Great presentation. Do you think in natural infections, you also get this mix of strains producing HARP-X or HARP-G?
So this is a question that Julien really like and try to study right now. So about the phenotypic heterogeneity in clonal population, it was shown that there seems to be heterogenetic phenotypy for quorum sensing in planta. It was already shown, and it was shown for Pseudomonas also. So Julien is looking to see if he can see heterogenetic phenotypic heterogeneity for the type 3 secretion system.
Thanks.
Actually, I'm going to ask a question that builds on that before I get back to the chat. So yeah, so for the fitness increase, what do you speculate is that increase? Is it because the burden of making type 3 secretion is missing, or is it that the machinery is gone and they change their behavior completely? Like, what do you speculate is happening?
It could be both. I mean, for example, we know, and we discussed that with Julien yesterday, I think. We know that when you produce a type 3 secretion system, normally you don't produce the flagella. So does this mutant are motile? Apparently, yes. So is there an equilibrium between different systems, but you still have a bit of the gain of fitness you have is still enough to be observed? I don't know. I don't know if it's only the type 3 secretion system or all the physiological state of the bacterium change.
And then to the kind of the last questions in the chat here, and I think they're related. So does the performance of mutated bacteria differ depending on the age of the host? Does this have an effect on the type of genes that become relevant for fitness? So does age affect the host? And then someone in the chat said that we have indications that flower infections play a major role in the occurrence of seed infections. Do you also use RBT and seek after flower inoculation?
So 3 good questions. So the age of the host, I don't know, because we didn't test our fitness analysis on different old plants. So we did it on 4 weeks old cauliflower leaf. But maybe if you do it on younger leaf or older leaf, you will have different, I guess you will have, the physiology of the plant could be different, so it could affect the fitness of the bacteria. The other question was about...
So one was about age and one was about the flower.
The flower, oh yes. It will be really difficult to do RBT and SEC on flower colonization, because from what I know, you have really maybe 1 or 2 cells that will enter for seed colonization, so it will be really, really challenging. But if you have a huge greenhouse, maybe you can, but we cannot do that. Thank you.
Okay, thank you, Alice.
Thank you very much.
So you might tell from the schedule we're a little delayed, but that's because one of the presenters, their presentation wasn't uploaded. So we're actually going to be missing one of the talks here, and then we also were able to do we're able to speak in memory of our colleagues. So our next speaker is Chiara BERNARDINI, and she'll be presenting on the role of phloem membrane contact sites on Candidatus liberibacter asiaticus infection.
Good morning to everyone. I'm thankful to the organizer to give me the opportunity to speak to you today. I'm Chiara BERNARDINI. I'm going to talk about the role of a membrane contact site on Candidatus liberibacter asiaticus infection. First of all, we need to focus our attention on the players of this topic. So what are membrane contact sites? Membrane contact sites are areas in which two membranes that they belong to different organelles, they come in really close contact without fusing. These areas are characterized by high vesiculation, high lipid exchange, and basically they are a net formed by three proteins, VAP27, NITRC, and synaptotagmin. We always wonder if membrane contact site can have a role in the infection in general. And has been reported that synaptotagmin A can have a role in the viral infection. As you can see in this model, synaptotagmin A can interact with the viral movement protein to improve the movement of the virus, increasing the plasmodesmal gating, but also can take place in the formation of the viral replication complex. And was also demonstrated that the downregulation in Nicotiana bentamiana and in Arabidopsis of synaptotagmin A, that I will call hereafter CETA, can delay the symptoms appearance. We focus our attention on Huanbin. Huanbin is a really important disease in Florida. If you think that in the last 15 years, the production reduced from 100% to 20%, Huanbin is a pretty big deal. And it caused important quality and quantity losses because the leaf system of the tree decay and the death of the tree occur within some years from the infection. The putative causal agent of Huanbin, putative because we are still not able to culture him in vitro, is Candidatus liberibacter asiaticus, and is a gram-negative bacterium and phloem restricted. So when we start to study cellulose, we wonder what is this attachment structure between the cellulose and the membrane, and we start to wonder if can be a special kind of membrane contact site. So the main goal of my project is understand if synaptotagmin A can have a role in the phloem bacterial infection. But to do this, we firstly need to demonstrate other two secondary objectives. The first one is if CETA localizes in the phloem, that is the site of infection of the pathogen, because until now was not demonstrated if CETA was also inside the phloem, and specifically inside the C element. The second objective is if the synaptotagmin is modulated during the infection. Then, after demonstrating these two objectives, we can move to the third one and demonstrated membrane contact site and synaptotagmin had a role in the bacterial infection. So about the first objective, the localization of CETA, I cross a SZUREK GFP line that has GFP expressed only inside the phloem, because SZUREK is the sucrose transporter responsible for load and unload the sucrose inside the phloem, with a CETA RFP line that has CETA attached to the RFP and is expressed everywhere inside the plant. So we cross it, and we obtain the crossed plant, and we observe with a confocal little plant around 20 days old to localize the signal. So as you can see, here I report only the crossed plant. In the first column, you can see the signal for the SZUREK GFP that is only inside the phloem. In the second column is the signal of the CETA RFP that is ubiquitous, but it seems that has a pretty high concentration in the phloem. And from the merge, we were able to say that CETA co-localize in the phloem with SZUREK. So after that, after this work that we did on Arabidopsis, we move on our pathosystem in orange. It's important to know that in citrus, CETA has two isoforms, X1 and X2, and we start to think if these CETA isoforms are modulated during Candidatus liberibacter infection. So we collect healthy and infected plants from the field, And we extracted RNA, we ran the gene expression analysis, and together we also ran a Western blot. From this part, we can see that X1 and X2 are both involved in the infection, and the gene expression suggests us that we have a strongly upregulation of both the isoform, and if you see at this heat map, here I analyze the CT value to understand if one isoform is more important than the other, and it seems that X1 is the most concentrated isoform in all the plant. So we start to wonder if also the protein is higher, not only the transcript. So we perform a Western blot using an antibody for Arabidopsis, but that has a target site shared with CETA in citrus. And we show that also the protein increases in case of infection. So then we move to our third objective, and we took infected citrus plant, sweet orange plant, and we downregulated CETA in the phloem through virus-induced gene silencing, mediated by CTV. Citrus tristeza virus is a phloem virus, so it carries our construct only inside the phloem. So as a result, we had plants that are downregulated only inside the phloem. And we use CTV-GFP as a control group. On those plants, we perform symptoms evaluation, confocal observation, and gene expression analysis. So about the symptoms, we can see that in CTV-CETA plant, we have less symptomatic flashes than in CTV-GFP plant. But also, what was really interesting was the symptom severity that was reduced with the downregulation of the CETA. So here you can see that the green line is less symptom severity. So not only we had less symptomatic flashes, but also with less important severe symptoms. So we start to think that maybe this result could be caused by an enhanced callus deposition. In fact, it was already demonstrated that in citrus, the presence of psilas increased the callus deposit inside the phloem. So we took the picture of our plant with the confocal, and I analyzed the picture with the Fiji, and we obtained the number and the area of callus deposits. And what was surprising was that the number of callus deposit was higher in CTV-CETA plant, but the total area was lower in CTV-CETA plants compared to CTV-GFP plants. Also, we analyzed the gene expression of the two genes involved with the phloem plugging, Cal7, that is responsible for the position of callus at the sieve plate, and PP2, that is responsible for the production of the phloem protein at the sieve pore level. And both of them, they were downregulated in case of CTV-CETA plant. I'm running out of the time. I will go quick on this. We try to see in Arabidopsis if the callus mechanism was downregulated without any infection. And so we took the picture of the epidermis of CETA plant, that they have a downregulated CETA in all the plant, and we discovered that the count of the callus deposit was lower, and also the area of the callus deposit was lower. So, in conclusion, we can say that the membrane contact site has a role in the phloem infection, because in a CETA downregulated plant, we saw an altered callus metabolism and less symptoms. And we explained the altered callus metabolism with a disturbance of the calcium signal inside the plant. In fact, CETA has some binding site for the calcium. So what we suppose is that CETA can subtract calcium in the lumen of the sieve element and cause an altered signal for the callus deposition. Also, as I mentioned before, callus is one of the main reasons for the symptoms appearance. So it's possible that this altered callus alter directly the symptomatology. Another important thing about the symptomatology is that the disruption of the membrane contact sites could affect the bacterial replication. And also, we suppose that could exist a steric interaction between the bacteria and the seta that could affect the movement. So I will leave you with this take-home message that the bacteria has, the membrane contact site has a role in the phloem bacterial infection, and about the mechanism, we are still working, and I hope to give you something more information in 2028. So with this, I want to thank you for your attention, and I want to thank my lab for the support during this study and my funding agency. Thank you so much.
We have time for a couple questions, but to get it started, have you quantified CLAS with qPCR or only evaluated symptoms in the sweet orange transform plant?
We quantify, and the difference in the titer is not significant, and so we suppose that is more that the CETA affect more the distribution of the pathogen instead of the replication of the pathogen.
Any question from the audience?
Well, I think this might be a good opportunity to keep on moving forward the program because Dr. Zhou uploaded his presentation, but that will push his all the way to the end to let the other speakers that had theirs uploaded on time present, and then we'll move forward with this.
So thank you very much again.
Thank you.
Yeah.
Thank you.
And let's move to the next one. So our next speaker is Yael HELMAN from the Ruhr-Universität of Jerusalem, and just let us give you the floor. You want some?
So, hi everyone. So thank you, Jonathan and Boris. It is a huge venue. I agree with Alice. I feel like Beyonce, but luckily for you, I won't sing. So in our lab, we study microbial interactions, and as part of this, we are interested in how volatiles mediate such interactions. And what we do, we screen for active volatiles using different methods, looking at single-cell morphology, colony patterns, transcriptomic, proteomics, and many more. And today I'll tell you a story about the interaction between two groups of bacteria. The first group is Penibacillus bacteria. These are model bacteria that we study in the lab. And one very important thing about them is that these are very proficient swarmers. That means that they can swarm on 1.5% agar and more. Another interesting thing is that they are very sensitive to the environmental conditions. So they change the patterns that they make according to the environmental condition, which makes them really good candidates to study microbial interaction. Another relevant characteristic of these bacteria to this talk is that we found that their swarming behavior is subjected to carbon catabolite repression, meaning that when we grow them on glucose, as you will see here, they grow really nicely, but they don't swarm, whereas if we grow them on LB plates without glucose, they grow and swarm. That means that there's a downregulation of swarming-related genes once we grow them on glucose. The other group that's I'm going to talk about in these interactions are Xanthomonas bacteria, and I will use Xanthomonas perforans as a model organism. As you probably all know, this is a plant pathogenic bacteria. It causes bacterial spot disease in tomatoes and peppers. And what's really relevant to this talk is that although it has a flagella, it cannot move on solid surfaces, and it's dependent on water in order to reach openings on the plant and infect. A former postdoc in the lab, she just finished, Darni, a very talented postdoc in the lab, found out that volatiles of Xanthomonas perforans can override the glucose inhibition that I showed you before. So what you see here are two divided Petri dishes. They have a plastic barrier between the two compartments, meaning that only volatiles can transfer from one compartment to the other. In the top compartment, you have LB with glucose, and you can see that you have 3 colonies of Penibacillus in this plate, in the left plate, and they are growing really nicely, but they are not swarming. On the other plant, we put Xanthomonas in the lower compartment, and what you can see is that we do see swarming, and we can quantify this and just look at the swarming area, and you can see that it increases. And when we look at the transcription of flagella-related genes, which are downregulated in the presence of glucose, when we add Xanthomonas, you can see an upregulation of this gene, meaning that volatiles of Xanthomonas can override glucose inhibition, which was pretty surprising for us, because what we know about the CCR is that it usually works through the PTS, through the phosphotransfer system, meaning that when glucose passes through the membrane, it gets phosphorylated, and there's a phosphorylation cascade that usually results in the inhibition of gene transcription, and this is true both for gram-positive and gram-negative bacteria. So we are thinking, how can volatiles of one bacteria change the outcome of this whole pathway? And we started looking at the literature, and we found this really cool work by you et al. That was published already 10 years ago. And what they showed is that the nitrogen to carbon influx into the cell can influence the CCR outcome. And they show that this can happen through changes in the level of specific precursors that you can find in the intersection between carbon metabolism and nitrogen metabolism. And when we thought of what type of precursors this can be, we immediately thought of alpha-ketoglutarate, which is in the intersection between the TCA cycle and the GOGAT pathway, which is carbon and nitrogen metabolism. And it's been known for many years that intercellular levels of alpha-ketoglutarate can fluctuate according to the carbon and nitrogen availability in the environment. So what we thought, maybe volatiles of Xanthomonas can override the glucose inhibition by changing the carbon to nitrogen ratio in the environment. And if this is true, then most likely these volatiles carry an amine residue. So in order to check if this is true, we collaborated with a long-time friend, Stefan Schulz. He's a chemist from the Technical University in Braunschweig. He's, I think, one of the world-leading experts studying bacterial volatiles. And what they did in his lab is they used this test where you put in one substance, and according to and if there's an amine, and according to the amine, you get a different substance. In this instance, we got isobutyl carbamate, which indicates that there are high levels of ammonia in the headspace of Xanthomonas. So our next step was to check whether we can get the same reaction that we got with Xanthomonas, but with ammonia, and we did. So as you can see here in the plates, instead of putting Xanthomonas in the lower compartment, we put ammonia, and we got exactly the same response that we got with Xanthomonas, meaning induced swarming and overexpression of flagella-related gene. That means that the ammonia produced by Xanthomonas overrides the glucose inhibition, which was really interesting for us because we work a lot on microbial motility, and we learned a lot just by looking at this interaction. But since you all know, and we know that motility is extremely important for plant infection, we wanted to see whether this type of interaction can have an effect on plant disease. And here I want to put in another piece for to the puzzle, is that I showed you that Xanthomonas can override glucose inhibition, but we also found that volatiles of Xanthomonas can actually induce swarming initiation in Penibacillus bacteria even without the presence of glucose. When you take bacteria from liquid media and you put them on solid media, especially proficient swarmers such as Penibacillus, they need to elongate and they need to hyperflagellate it. And when we put these two bacteria together, we saw that when we have Xanthomonas in the area, they start swarming sooner. So we used two different techniques to measure why, and one device is called an image stream flow cytometer, which is very similar to a flow cytometer, but every event that passes through the detector is also being imaged. So we can count the cells, but we can also measure their length. And what we saw is that when we expose Penibacillus to volatiles of Xanthomonas, they start to elongate much sooner. We also did some transcriptome analysis, and we saw that there's an increase in flagella-related gene, meaning that volatiles of Xanthomonas can act as signals that promote differentiation of swimmer to swarmer cells in Penibacillus. And when we put these two bacteria together, not in a divided Petri dish, this early induction of swarming actually creates a directional motility of Penibacillus bacteria towards Xanthomonas, which is stuck there on the plate. It cannot move. So you see this white cloud on the plate moving towards Xanthomonas. But when we leave these two plates on the bench, and we were really lucky that Xanthomonas was yellow, we were surprised to see that after a few days, Xanthomonas started spreading all over the plate, and we could really see it stuck in vortexes of this Penibacillus. This specific Penibacillus is Penibacillus vortex. It moves in vortices, and you can really see the yellow bacteria stuck in these vortices. After 60 hours and after 90 hours, we could see it spread all over the plate. The scanning electron microscope, you can see the long rafts of Penibacillus beneath and fat Xanthomonas hitchhiking on top of them. And the cool thing is when we looked at these interactions on leaves and we used fluorescently stained Xanthomonas cells, we saw that we put two colonies, two cultures, one XP is Xanthomonas, PV is Penibacillus vortex. We saw that after a few days, this fluorescence colony of Xanthomonas starts spreading between the two plates, the two colonies, sorry, and we could really see it stuck in the vortices exactly like I showed you before in the plate. So Xanthomonas can hitchhike on Penibacillus both on plates and on leaves. Of course, when we put Xanthomonas alone, we didn't see the spread in fluorescence. So we wanted to see what happens if we co-inoculate these two bacteria on tomato plants. So we inoculated tomato plants either with Xanthomonas or with Xanthomonas perforans and Penibacillus YH6, which is a Penibacillus that we isolated from tomato leaves. This Penibacillus hardly moves, but when we put Xanthomonas next to it, it starts swarming massively towards the Xanthomonas. This is how we actually isolated it. We put a tomato leaf on a Petri dish, Xanthomonas on the other side, and it just lured the Penibacillus. And we looked at, when we inoculated these two on the plates, we looked at disease indices. This work was carried out by a very talented PhD student in the lab in collaboration with Saul. They developed this way to quantify disease indexes using machine learning, based on machine learning. And she measured the disease indices in tomato plants. She used relatively old tomato plants where it's hard to cause disease, about 6 weeks. And as you can see in the pink column, we could hardly get disease alone, but when we inoculated these two colonies, we got a significantly increase in disease indexes, which we found really interesting. So this is one type of interaction, but we wanted to see what happens if we put another player in the game, and this is Penibacillus dendritiformis, another model organism in the lab, that just by looking at its genome, we found a lot of polyketid, not a lot, but 3 huge clusters of polyketid syntase or non-ribosomal peptide synthetase that we speculate that they are involved in antibiotic production, and in collaboration with David Sherman from Michigan University, it seems, at least in silico, that these are several unique antibiotic compounds, and AD indeed showed that this Penibacillus inhibits the growth of Xanthomonas in vitro. And she did exactly the same test experiment as I showed you before, but instead of using Y86, she used Dendridiformis, and this time she used younger tomato plants, which is much more easier to infect. And as you can see here in the pink column, which is just Xanthomonas, we had got higher disease indexes, but when we put the two bacteria together, the two colonies together, you could see a significant decrease in disease indices. So basically what we're saying, which is not surprising, but it's nice to see, is that interspecies interaction has a significant effect on the disease that will develop later, but it depends who are the players. So if this the gray bacteria here are Penibacillus Y86, they are attracted to Xanthomonas by the production of ammonia. They start moving towards it, they carry it with them on the plant, and they might aid Xanthomonas reach openings on the plant, and this will induce disease. But if this player, instead of Penibacillus Y86, is Xanthomonas, then the disease will be decreased, and the amount of Xanthomonas will also, of course, be decreased. And now we want to do the same thing with a flagellar mutant of Penibacillus, and to see whether this hitchhiking is really the cause for it. And with this, I would like to, of course, thank the people who did the work, starting with Efrat, a former PhD student in the lab who started looking at the swarming induction by Xanthomonas, Dani, the PhD, the postdoc, and Adi, that does all the disease indices, and now she's putting a lot of efforts in discovering these antibiotic compounds, which at least preliminary experiments show that they can even work against MRSA, so it's not related to plant, but it's promising. And of course, Saul, who I love working with, and Sivan helped us do the machine-based learning quantification of disease, and Stefan. That's it.
Thank you very much, Yael. So before we move to the question in the chat, if there is any from the audience, I see Cindy. Yeah.
Yes, thank you, Yael. I always like listening to your talks. Plants produce volatiles. Do you know of the effect of plants themselves? Can they induce swarming at all?
We are checking. I didn't tell you the whole story. Well, it seems like the ammonia and the C/N ratio is the cause, but we actually found out that it's more related to pH levels, which is totally new, how pH can be signaling a molecule different in pH. But we are very interested in how different plants do it, and we are looking into it. I won't be surprised, because you can see bacteria, especially these proficient swarmers, they run towards plants. So yeah, for sure they attract them.
Okay, so some questions from the chat. One is, have you checked the effect of pH on Penibacillus swarming? Because ammonia may alkalinize the growth medium.
I'm not sure I understand. How do we can check the effect of pH either by manipulating the pH ourselves. We can use different buffers. We can measure the changes in pH when we expose them to different bacteria, and maybe I answered Cindy when I said that it's not the ammonia, but it's the pH, which was very surprising, and we are now putting a lot of efforts to understand how pH shifts affect swarming bacteria. So pH has a huge effect here, and we are looking into it.
Okay. Another question is, do you think that there is any benefits for Penibacillum in this interaction?
Yeah, that's a good question. There could be, because the minute Xanthomonas, let me restart. It can be just a chemical manipulation. It can be just a byproduct of this thing. Another option is that, at least for Y86, if it carries Xanthomonas and Xanthomonas infects the plant, it might provide more nutrients just by breaking the cells of the plant. And we are now trying to measure the growth of at least the cell number of Penibacillus together with Xanthomonas and without Xanthomonas, and seeing whether there's a benefit, at least for the Y86.
Maybe a last question. Have you screened for phytopathogens other than Xanthomonas perforans and find other that have the same effect on Penibacillus?
Yes, and we have a really big project together with Saul now, and Adi is working on it, on Acidovorax. Acidovorax citrulli, we get very similar effects, both with, especially with the Penibacillus dendritiformis, who is attracted to it but also kills it, or at least harms it, so we are looking into it, but yes.
Okay. Thank you very much, Yael.
Okay, our next presenter is Suayb USTUN, and it's about how proteostasis shapes plant-bacteria interactions.
Thanks a lot, Jonathan, for introducing me, and thanks also for both of you for inviting me. I'm really excited to be here. It's my first time at the ICPP, and I will take you a bit on an adventure on proteostasis, because this is the major research focus in our lab, and it's a bit maybe far away from pathology, but I will try to convince you that actually what we identified might be really important when we look into very general mechanisms like proteostasis and photosynthesis. So if you talk, when we talk about protein homeostasis, then you can basically think about it as the life and death of proteins. Do you see the pointer? Yeah. And this is basically the central dogma of molecular biology, that you have transcription, translation, and you have a protein, and then in the end it's degraded. And of course, it's like a very central mechanism that cells have, but we have to know that the tight equilibrium of biosynthesis and degradation of proteins is really important to actually react to sudden changes in the environment, like pathogen infection. And if you have a perturbation in this equilibrium between protein synthesis and degradation, this will cause usually to an aberrant stress response. So sometimes stress responses can be too harsh and lead to cell death, but sometimes also stress responses can completely not occur anymore. And I don't have to tell you why it's really important to study plant microbe or plant bacteria interactions. I think that everybody in the audience knows this. And one of our aims, of course, is to have resistance and to ensure food security. But you can also see it a bit from the other side. We can use these very clever bugs, the microbes actually, as tools to understand cellular pathways and identify novel immunity hubs that might be utilized to actually, again, then go back and have more resistance, or understand also like how the disease is occurring. And this is basically what we in the beginning initially did. And here we use a lot effector proteins from bacteria as tools to understand, and we identified, and also others identified, that the proteasome and autophagy seem to be major hubs where effectors, that effectors target to cause disease or to also inhibit disease responses. And this is also highlighted, for example, here. You can see when you look only into the ubiquitin proteasome system that bacterial effectors can target basically the system at various steps, which you can see here. So our research idea centers a bit around the idea that synthesis of proteins, sorting and degradation are tightly interconnected, and that this interconnection of these three major pathways has a central role in immunity and disease. And we have various projects, I will not go into this in detail, on all of these different aspects, and I just want to highlight three of them because we have three posters available by Ophélie, by Margot, and Manuel, and they are happy to share their new insights and their different projects during the poster session. So visit their posters today, please. They will be really happy to discuss their research with you. So today I'm going to talk about something that has been a story for 4 years. It's Gautier's PhD project, and it's concentrating on the proteasome. So the proteasome, you know, that is basically a very conserved degradation mechanism. It can degrade multiple substrates, cytosolic proteins, but also proteins from different compartments. Nuclear proteins, mitochondrial proteins, and also chloroplastic proteins and proteins that sit at the ER. And I don't have to tell you that, of course, a central degradation mechanism like this is really important in different cellular processes, and of course also in immunity. Several groups have identified proteins involved in perception or hormone synthesis and transcription factors that are degraded or recycled by the proteasome. So actually how we bumped into the proteasome was when we look into the Arabidopsis Pseudomonas interaction. And what happens here is that Pseudomonas is able to activate another degradation pathway, autophagy, and basically eats up the proteasomes and degrades them. In the end, it leads to an inhibition of the activity or suppression of the activity of the proteasome, which you can see here. So the wild-type bacterium severely suppresses the activity. But interestingly enough, when we look into transcriptional data, proteasome subunit genes seem to be transcriptionally activated. So we were a bit puzzled, like, okay, we have less activity, but more transcripts available. So we got a bit interested in how actually the expression of the proteasome megacomplex is transcriptionally regulated. And here we know a lot in yeast and in animals. So basically, proteasome activators here, they are undergoing a negative feedback loop. It means under normal conditions, they are constitutively degraded by the proteasome. But then when a stress occurs, which can be also a pathogen infection in these systems, the transcription factors are not degraded anymore. They accumulate. They can then translocate into the nucleus and drive proteasome gene expression. In plants, we actually know the two transcription factors that activate the proteasome stress regulant genes, but intriguingly enough, they are localized at the ER. So they have to be cleaved upon a stress, and then they are transferred to the nucleus. But the exact mechanism how they do this, and also their role in immunity, was so far not described. So what we thought we would like to do is, I will skip this slide here, that we subject our double mutant, because they act as a dimer, the NAC53 and 78, we subject our double mutant to several proteotoxic stresses that are that can occur. So we can use chemicals like BTZ that inhibits the proteasome specifically, and you can see the mutant is hypersensitive towards this. We can also use bacterial infection because the bacteria is suppressing proteasome activity, and here in a bacterial growth assay you can see only the double mutant is hypersusceptible towards Pseudomonas, while the single mutants, they don't have a growth effect. They grow like Columbia 0. Like in Columbia 0. And then we basically probed different organelles, because you can also perturb or induce proteotoxic stress by perturbing, for example, translation in chloroplast and mitochondria, but also inducing ER stress. And all of these different stresses resulted in a modified response of the transcription factors, but I will come to this at the end of the talk, how actually it's modified, because it's a bit different than what we observed here. Nevertheless, we concluded that generally proteotoxic stress is inducing, or that the transcription factors are really essential during proteotoxic stress. But we actually wanted to know like how they integrate various the signals from various cellular compartments, because we actually look into several compartments here, and we wanted to know how they integrate this. And knowing also that they are both localized at the ER membranes, you can see this here in confocal images in this reticulate structures that co-localize with an ER marker, we kind of wanted to see, okay, what is the proteome or the interactome of both transcription factors in a steady state situation, but also during an infection where we cause a malfunctioning of the proteasome. So after several rounds of proteomics, we came up with a core interactome. We have actually much more interaction partners, but here I will just reduce it to the most prominent ones. And I will also just talk today about actually the proteasome and the ERAD components. So ERAD is ER-associated degradation. So basically proteasome degradation that happens at the ER, and we identified key components of this pathway as putative interactors. And we also verified these interactions with these components with our NAC transcription factors. And I want to just highlight one of these components, which is the HRD1 A and B E3 ligase complex. And basically by confirming the interaction, we also kind of wanted to know, okay, are they really targeted by ERAD? And the idea came actually when Gauthier made his first transgenic lines in the lab. So he kind of fused the transcription factors to GFP and generated Arabidopsis lines, and he could never see a fluorescent signal. But when we treat these plants with a proteasome inhibitor, you can actually nicely see suddenly the signal is appearing in the nucleus. And this is also true when you look into the protein levels. The protein amount is severely increased, especially upon Pseudomonas infection. So knowing this and also like having in mind the proteomics data, we thought, okay, we will look into the ERAD pathway. And you can see here the pathway very simplified. Basically what happens at the ER that this E3 ligase complex, HRD1A and B, it ubiquitinates substrates, and then via the help of this unfoldase CDC48, proteins can be degraded via the proteasome. And we kind of hypothesized that the NAC transcription factors are degraded by this pathway. The cool thing is that we can inhibit the pathway by using very specific drugs. So there is a drug for all of these different steps available. Doing then biochemical assays, but also confocal microscopy, we kind of see that each time when we inhibit one of the components of the pathways, we recover basically the signal. So this tunicomycin treatment here is basically just a negative control. So knowing this now, we kind of could confirm that indeed the NAC transcription factors in a normal condition are degraded via the ERAD pathway in plants. And this is just to show you, I will not go into detail because I also don't have so much time, that the E3 ligase complex really directly ubiquitinates the transcription factors, as we also identified in vitro and also in vivo several ubiquitination sites in the transcription factors. So this is just a visual summary again here that the transcription factors in a normal condition are degraded via ERAD. And cool enough is that although the transcription factors in animals and in plants are very evolutionary distinct, so they have no relation to each other, the pathway is actually conserved in animals, because here also in a normal steady state condition, the transcription factor is degraded via the ERAD pathway. So in this first part, I basically showed you there is some kind of a negative feedback loop also in plants existing. So under normal conditions, the transcription factors are unstable and degraded, and just upon stress, they're stabilized. And of course, when we talk about stress, we then looked back into our pathogen infection, because we wanted to know actually why our mutant is really hypersusceptible here. Is this really proteasome related? Because the proteasome genes are not upregulated anymore, which would be a bit counterintuitive to what we have found before. And we also looked into some classical immunity outputs in our double mutant, and we didn't find anything really super striking. So the mutant basically behaves like wild type when it comes to the immunity outputs. So we then thought, okay, we would like to identify other targets of these NAC transcription factors, and conducted an RNA-seq experiment comparing our NAC mutant to Columbia 0 in an infected situation. And in the heat map, you can see here the last column is the most important one. There is a small cluster here that seems to be repressed, and this small cluster is actually the proteasome megacomplex. So all of the proteasome genes, they cannot be activated when the NAC transcription factors are not present during infection, which is cool because basically it confirms that the Pseudomonas bacterium is inducing a proteotoxic stress. But then the rest of the differentially expressed genes were all seem to be activated in a mutant, which means they might be candidates where the NAC transcription factors might repress them. And this is also just to show you that the differentially regulated genes, they are genes from different pathways. You can see auxin, cell wall, trafficking pathway genes are affected. And then one cluster stick out a bit, which was the photosynthesis cluster. So what Gautier then did, he basically looked into the promoter regions of the different genes, because we know the promoter motif of the proteasome genes where the NAC transcription factors bind. And what he identified is that only in the photosynthesis-related genes, he can actually see that the promoter motif is also present where the NAC transcription factors bind. So it's this TGGC motif. We verified the direct interaction of the transcription factor by using EMSA, which you can see here. We took 2 candidate genes from this photosynthesis genes, so our transcription factors, they can bind to it. We have done also several controls by mutating the motif, so then it doesn't bind anymore, so it's really directed via this motif here. And a very cool experiment, what he did is, in vivo, he did chip PCR during infection, and only during infection he can actually pull down these promoter regions from these photosynthesis-associated genes. So apparently during infection there is a specific binding of both transcription factors to these photosynthesis genes. But actually what happens to them? So we used first a reporter assay here to show that both transcription factors can repress the activity of these photosynthesis-associated nuclear genes. And then when we look into a more physiological output, when we measure photosystem 2 activity, you can actually see that usually, of course, Pseudomonas infection, like a lot of other pathogens, reduces the photosynthesis capacity, but in the mutant it's attenuated, which you can see in the measurement, but also in the pictures here. So it looks like that we have a bit some kind of a trade-off here, because the infection, it causes a suppression of the proteasome, but the suppression activates the NAC transcription factors, because of course you want to have a new proteasome to deal with the proteotoxic stress. But additionally, the transcription factors, and this is something that we don't know if this is really in parallel happening or if there are some co-components needed for this, but in parallel, I would say photosynthesis gene expression is repressed. And because both pathways are very fundamental, we thought, okay, is this like a general phenomenon? Does this happen in all kinds of different stresses? So what Gautier did, he took 170 RNA-seq conditions that are publicly available and that are published, and he kind of correlated the expression pattern of proteasome genes to photosynthesis-associated nuclear genes. And what he basically sees is that this seems to be a general phenomenon. So if you have a biotic stress, abiotic stress, hormone treatment, you have proteasome activation, but photosynthesis repression. So there is a negative correlation of both factors in the majority of the stresses that we looked into in this 170 RNA-seq conditions. And this brings me also to my summary slides. So I told you, like in steady state, both of our transcription factors are very unstable. They are degraded by the ERAD pathway. But then when we have a proteotoxic stress, which can be an infection, and this can be bacterial infection, but also infections by fungi, that the transcription factors are released from the ER, a membrane. They are not degraded anymore because the proteasome has too much other things to do and is suppressed in its activity. And then they bind to the proteasome genes and photosynthesis genes. And they can repress the photosynthesis genes to dampen photosynthesis activity. And we think that these NACs, they mediate the communication between the nucleus and the chloroplast because they seem to have like a broader role in different kind of stresses. And this brings me to my initial slide where I talked about proteotoxic stress and the different compartments, because if you look into these substances or antibiotics that inhibit translation, they usually dampen the PS2 activity, the photosynthesis activity. But the NAC transcription factors, they can actually perform a bit better in these conditions, because of course they are not there to repress the photosynthesis genes anymore. And we think that maybe they have a role in this retrograde signaling of nucleus and chloroplast. And just to put this in a small perspective, in humans, actually, the human counterparts of the NACs, they have a role in mitochondrial biogenesis, and we have actually some exciting new in silico results where we see that also in the mitochondrial biogenesis genes in plants, the motif where the NACs binds is conserved. Yeah, and this is just a cartoon basically depicting, although we have these distinct, evolutionary distinct transcription factors that they're all there to cope with proteotoxicity, and we hypothesize they might be all there to repress or modulate energetic metabolism. And with that, I'm at the end, and I want to, like, my biggest thanks, of course, are for Gauthier, because he did all of this work within 4 years, and currently he's writing his PhD. That's why he's not here today. Of course, also facilities, collaborators, funding, and you for your attention, and I'm happy to take questions. Thanks.
We already have some questions in the chat, and then if you have a question, please come up here. The first one was great talk, Suayb. Thanks. Do you know if these NACs work on their own or form different complexes depending on the type of stress?
I guess they mean the NAC transcription factors. That's a good question. I mean, they work as a heterodimer, the NAC 53 and 78, but we have indications that the network might be much bigger because we have other NAC transcription factors that might interact, and they might, I mean, we hypothesize maybe they have a role when it comes to the mitochondrial biogenesis genes that we had. That's something that we will maybe explore in the future to see, yeah. And also about the repressor activity of the NAC transcription factors, maybe they still need a co-factor, a co-repressor. And we have some candidates also in the proteomics identified, yeah.
Here is a question for you. So in your case, do you know how is the ER-associated degradation is activated or initiated?
So I think the activation and initiation is, I'm not really sure if I understood the question correctly.
I mean, how the ER-associated degradation is initiated?
I think that's a good question. I guess that because at steady state, because they're so unstable, it's basically active all the time. I mean, when we look into the proteomics, there are some proteins that might play a role in this activation, but usually the activation of the ERAT is actually through the ubiquitination of the substrate by the HRD1 complex. But how the complex is activated, this, I don't know. It's a good point, yeah.
Okay,
thank you.
And we have two questions left in the chat. So how do NACs have repression and activation activity when binding to the same motif?
That's an excellent question. I've been discussing this a lot with Gauthier. We have no idea. And I think maybe maybe there is something else, a co-repressor or something like this that somehow helps, but we don't know this. It's a good question, yes.
Alvaro asked it, so if you want to ask somebody that asked it, Alvaro asked it, so you can chat with him after. The last one is from Sichang Wang, and actually this is one of the questions I had. So how does PST involved in this process? Directly by type 3 effectors or indirectly through plant immunity? And I guess to follow that, I was curious, is there are steps in this where in the process of immunity and recognition or suppression is this happening?
So I think that because PSG directly targets the proteasome, I think that this mechanism, basically that you would suppress photosynthesis, is more coming from the plant, like when it senses, okay, something happens to the proteasome, and then basically these transcription factors are also used to dampen photosynthesis and try to kill maybe the tissue that is infected. This is one possibility, but then I also think, maybe the proteasome just acts as a trap to activate this immunity pathway, but maybe also there is a specific thing that the bacterium actually wants this. Because, there are really old papers showing that photosynthesis or sugars, metabolites that have antimicrobial activities, and maybe the bacterium wants to shut down photosynthesis. It's a bit like we don't know like what comes first a bit. Yeah, it's a good question.
Thank you.
Yeah, thanks a lot.
Yeah, so we will move to the next and final speaker for this session, Huanbin ZHOU from Beijing. And there is several questions in the chat that we could not address for today's session, so I would ask the speakers to answer them in the chat so that everyone can have the information if they want. Thank you.
Hello everyone. It's my great honor to be invited here to give this presentation. I'm going to share with you our project on the CRISPR-mediated genome editing in bacterial plant pathogen. So Bernard, before me just talked about tail effectors and the decay slide, I'm going to present the case 12 system and the non-tail effectors. So first, a little bit introduction to our lab. We're mainly working on the interaction between rice and the Xanthomonas rhizae, the bacterial blight causing pathogen, as well as rice molecular design. Different to the traditional ways, we introduced the CRISPR system, the technology into each part. So in the last 10 years, we developed lots of genome editing technology on rice, including a large number of case nuclease and base editors and prime editors, which allowed us to rapidly and easily generate the gene knockout, knock-in, knockdown, knock-up mutants, as well as novel rice germ plasmids. Just basically the CRISPR system technology just makes big difference in the rice basic studies and breedings. So this is the opinion paper we wrote last year for the molecular plants for celebrating the 20th anniversary of the draft sequence of the rice genome. So at that time, we are so excited to say the rice genome is so editable. But now we have more tools. So, so compared to the rice researchers, we are the CRISPR studies on bacteria, especially bacterial plant pathogen, next way behind. So these two methods, transposon insertion and homologous recombination, are widely used in the lab worldwide. But considering the great power of CRISPR technology, we are aimed to develop all the tools, CRISPR tools for the Xanthomonas rhizae, just like what we've done in rice. So first we need to find out a case nuclease. Actually, we all know the CRISPR system comes from the bacteria and the archaeon. Actually, our Xanthomonas rhizae, Passival rhizae, just has a native CRISPR Cas3 system. My supervisor, Dr. Yang, at Iowa State, and then his collaborators just demonstrate the self-editing by Cas3 nuclease in the PXO99A, just called cell death, which means there's the CRISPR system is active there. It cut the genome DNA, but the double-strand DNA break cannot be repaired. Later on, the genome ZHOU SNAP just revealed this Cas3 induced cleavage can be repaired by the heterologous recombinase, LIPME, when donor delivered. Okay, but compared to their research, we're more interested in the easy-to-use and versatile genome editing tools. in this case, no consideration of the presence or absence of the native CRISPR system, and the low consideration of donor templates. So we look at the whole CRISPR case system. There is one, the case 12, caught our attention because the case 12 is capable of processing the pre-CRY-RNA into mature RNA, which means case 12 system is really good at multiplex genome editing. This is the case 12 gene family. It's originally named as CPF1. So case 12 nucleus creates staggered cuts with 4 to 5 nucleotide overhang. So we choose the FN case 12 because this nucleus recognizes a really simple pen, TTN. Okay, so we designed an intermediate CRISPR vector for the FN case 12A. So the whole CRISPR cassette can be released and integrated into the other destination vector if needed. So the FN case 12 is under the control of an inducible promoter, just to reduce its potential toxicity. And the CRY-RNA is driven by a constitutive promoter. So we designed a guide RNA just for targeting the non-tail effector DOP-N in the PX099 and did transformation. We can see the transformation efficiency is really low. We just get on just a few colonies, and all those are active, which means the FN case 12 is active. It cut the genomic DNA, but the double-stranded bricks cannot be repaired. So I happened to read this paper. It showed the ectopic expression of the microbacterial tuberculosis KU and the like-ST protein can rejoin the DNA, the DNs of a linearized DNA in E. coli. Okay, so it gave us a clue. How about deliver these two proteins together with CRISPR system into the PX099 cells to see what it happened. Okay, so we updated our CRISPR construct with these two genes, and we repeated the experiments with the DOP-N CRY-RNA, and you can see the transformation efficiency just is higher compared to the former plasmids. Those colonies was randomly selected to PCR. You can see the gel picture. Those shift bands just means genome editing events occurred there. So a number of the colonies was genotyped by the single sequencing of PCR products and the whole genome sequencing. We can see the FN case 12 just induced the deletions in exon, ranging from several kilobase pair to several thousand kilobase pair. This is different to what we saw in rice. In rice, the FN the case 12 nucleus just induced indels, including both deletions and the insertions. And a micro-homologous region just at the breakpoint junctions were also detected. So, which suggests both non-homologous and joining pathway and micro-homologous mediated pathway just is involved in DNA repair when these two proteins used. So we also talked another non-tail effector gene, DOP-N. It gives us the same picture. So those shift bands and a similar ratio of different genome editing events. So next we like to see how about the F1 can K12A used in multiplex genome editing. At the first place, we just mix those ZOB-N, both ZOB-N and ZOB-V CRA plasmid. Just mix and the electrophorated into the PXO99 cells. But we only the single edited event at either ZOB-N or ZOB-V site can be detected. There's no double co-editing events. So we just assemble the both CRA together into a mini array because we know the FNK12 can process it into 2 mature CRA. So with this strategy, the co-editing efficiency is really high, but it's hard to find out a colony with small deletions at both target site. Okay, and then we tried the third strategy. We just used the 20 nucleotide spacer. Just short the spacer. Use the short spacer. It presumably just attenuated the nucleus activity of FNK12. So in timely repair can occur. So with this strategy, we can identify those colonies. With just identifying those this gene double mutants with the small deletions. So next, we tried the mini array to do the gene cluster deletion. This is the example. We designed 2 spacers to target both ends of this gene cluster. We can see just these 2 colonies, just this 3.4 kilobase per fragment were precisely deleted. So next question is, if the genomic editing is done, can you take out of the plasmid CRISPR constructs? So we went back to our the PHZ-B3 vector. We know that construct can cut the genome DNA, but the DNA break cannot be repaired. So we designed 2 spacers, one target the replicon of the plasmids, both plasmid ORI-V, and another one just targeting the legacy gene. So the plasmid curing of B4 plasmids and the self-targeting, self-curing of B3 plasmid can be achieved simultaneously. You can see the gel picture. The plasmid-free colonies is really easy to identify. Okay, so with the combined use of the B4 and the B3, 2 plasmid, we can do rounds of genome editing in Xanthomonas already. So PXO99 encodes 25 long-tail effectors, of which a number of DOP genes has been confirmed as the avirulence or virulence factors in the interaction between the rice and the Xanthomonas already. So we aim to just create non-tail-free strains, so which is important for the future study, studying the single long-tail effector. So those ZOP gene and the gene cluster was sequentially knocked out, and we got the D25E polymutant strain. So you can see here the efficiency is really high, and we tried lots of PAM sequence. Just in summary here, just the FNK12 just recognized TTN, TTV PAM in Xanthomonas already. Okay, so the D25E strain was subjected to whole genome sequencing for off-target analysis. Besides those those on-target mutation events in those 25 ZOP genes, five mutations, including 3 SNP and 1 insertion, 1 deletion, were identified in the whole genome. Okay, among them, only 1 SNP localized in the coding region, which means just considering of this polymutant just went through 42 times of FNK12-mediated editing, including both genome editing and plasmid curing. And these 5 mutants is independent of the is CRNA independent. So we presume the FNK12-mediated genome editing, just with stringently designed CRA, has no obverse off-target effects. And the rice seedling was challenged with by the D25E mutants. The deletion length is significantly shorter than the wild type, and slightly longer than the HACU mutant. Okay, just approximately 12,000 guidelines, just targeting all the genes in the PXO99 genome was designed and synthesized on microarray and construct. We detect 11,587 guidelines in the amplicon sequencing of the CRISPR lab. So this CRISPR lab is ready for the high throughput phenotypic assay in the future. We also tried this system in the Pseudomonas syringae to see if this system can be used. Okay. So the first guideline is designed to target the the effector gene HOP-AB2 on the chromosome. So you can see here we got the mutant, and another one. This strain has the DC3000 strain has 2 native plasmids. Okay. So in these experiments, we designed a spacer to target the HOP-X1 in the plasmid to see if we can take the plasmid out. You can see results. The plasmid is eliminated in all the 100% of the cells examined. So both constructs can be used in Pseudomonas syringae. Okay, I'd like to stop here. So thank you all for your attention.
Just because we're up against lunch right now, maybe one question. I actually have a question. So have you thought about an alternative approach where you would knock out, let's say, the pit box that's in front of most type 3 secreted effectors as a general approach to just knock down the expression of a bunch of effectors instead of a bunch of individuals? Have you thought of that approach, or targeting promoters rather than the gene coding sequence?
Sorry, I cannot catch your question. What's that? You want just
instead of targeting
gene cluster?
No, instead of targeting the coding sequence, just target a similar promoter motif for all type 3 effectors, like the
that's the CRISPR-I.
Yeah. No, but you can delete that promoter, right?
Yeah, sure. It's really easy to do that. You can just design 2 spacers to target the region. You just delete the region, really easy.
Okay, thank you for your presentation.
Okay, thank you.
And thank you everybody for coming and the non-conventional format. Please go support the posters that are related to plant-bacterial interactions, especially for the trainees here.
Thanks again. Bye.